Welcome to our presentation entitled Rubber Closures, Tests and Standards. Here is an illustration of all the rubber closure for parenteral preparations. We'll be covering some of these examples later on in our presentation. First of all, I would like to highlight the term rubber being a subset of elastomers. The properties of rubber is important for parenteral preparations as it can affect the physical, chemical, and biological stabilities of these preparations, which in turn can have an effect on the efficacy and toxicity profile of the drugs. We will be covering tests from USP, European Pharmacopoeia, BP, and JP. In addition, we will be highlighting some of the standards for elastomeric closures as stated by the ISO. We will be covering the tests and standards based on the properties of the rubber, namely physical-chemical properties, functional properties, and biological properties. Firstly, hardness is a significant property of rubber. It can affect the ease of penetration by the needle. The harder the rubber, the more difficult it is for the needle to penetrate. Hard rubbers tend to be more durable, but more difficult to mold into the shapes required for use as elastomeric closures. For example, injection points for flexible bags utilize softer rubber varieties to allow for easier penetration. Syringe plunger, however, uses the harder type of rubber to ensure smoother gliding through the syringe. There are two factors affecting hardness. Vulcanization is the process where sulfur is added to the rubber and heated during the manufacturing process. Vulcanization time therefore affects the hardness of the rubber. In addition, fillers are added to improve several properties of rubber, one of which is hardness. The hardness of the rubber can be determined by chemical tests. One popular method is the residue on ignition or evaporation. Upon heating, inorganic materials like the filler will remain. Organic materials such as the elastomer will be absent. Therefore, the mass of the residue can indirectly indicate the proportion of the fillers to the elastomer, the ratio of which determines hardness. This test is covered by the European Pharmacopoeia and JP. Essentially, there are three steps to this test. Firstly, the sample solution containing rubber is evaporated to dryness. Then the residue is dried and subsequently weighed. Physical tests can also be used to determine the hardness of the rubber. The equipment commonly used for these tests is the durometer and the IRHD hardness meter.
The ability of rubber closures to absorb moisture should also be evaluated. Rubber closures absorb moisture before it is fitted on the container. The absorbed moisture eventually enters the medicinal product in the container via desorption, and this is particularly problematic for lyophilized drugs, oily drugs, and drugs present in very small concentration. Such exposure to moisture compromises on such formulations' biointegrity, solubility, and concentration. Aqueous solutions, however, are less affected as the quantity of water that enters the solution is very small compared to the existing volume. Before fitting, rubber closures undergo steam sterilization, which introduce a significant quantity of water into the closures. The rubber closures must then be dried to a level suitable for drug application. For example, rubber closures for vials containing lyophilized drugs should have a lower residual water content than those for aqueous drugs. In the industrial setting, the closures are typically dried between 80 to 110 degrees Celsius for 1 to 16 hours. The moisture content of elastomeric closures can be measured with a standardized method called the colorimetric Coefficients titration. This method involves the use of electrical current and is mentioned in an ISO document. The ability of rubber closures to absorb preservatives should also be evaluated. Preservatives can partition into the rubber closures from the parenterals, thereby losing its efficacy in the formulation. The extent of absorption is affected by the type and concentration of the preservative and the type of rubber compounds used in the closure. However, existing literature does not define a set of tests or standards to assess this property. The gas permeability of rubber closures is also an area of concern. Generally, low gas permeability is desired, especially for vial stoppers and prefilled syringes containing powdered and lyophilized drugs. This is to reduce the oxygen and water exposure. Such exposure will compromise on its biological integrity and shelf life. However, in some cases, a high gas permeability is preferred. Such is the case of the needle shield of the prefilled syringe. The needle shields are pre-assembled with the syringes and terminally sterilized by ethylene oxide. Ethylene oxide must be able to permeate the needle shield to reach the needle beneath. Gas permeability of the rubber closures should adhere to the standard of ISO 2782. In addition, pharmaceutical rubbers should also adhere to this ASTM standard for oxygen and water vapor permeability respectively.
Another important component that needs to be tested for are the extractables in rubber closures. Extractables are to be differentiated from leachables. Extractables are chemical compounds that are extracted from the container closure system using stronger solvents and/or a higher temperature than that during the actual use. While leachables are chemical compounds that are extracted from the same container closure system using solvents and temperatures that are representative of that during the actual use. Firstly, solution S has to be prepared. This would be the solution that is to be subjected to various tests for the extractables of rubber closures, and these are the steps that would be involved. There are 8 main tests that can be conducted. Firstly, the color and turbidity of the appearance of the solution are to be tested by visual comparison or a suitable ratio turbidimeter against reference suspensions. Next, the acidity and alkalinity are tested for using bromothymol blue solution. UV spectrophotometry at wavelengths of 220 to 360 nanometers is used to test for the absorbance of the extractables. Reducing substances are also tested for using titration involving the reduction of potassium permanganate and sodium thiosulfate as the reagents. We test for the concentration of ammonium by color comparison with the ammonium standard solution. Zinc is tested for using atomic absorption spectrophotometry with the limit being 5 ppm. The heavy metal that is usually tested for is lead. However, an additional test for cadmium is also required for by JP. Lastly, the limit of volatile sulfides are also tested for in rubber closures. This is significant as sulfur is involved in vulcanization in the manufacture of rubber closures. The intensity of the black stain on the lead acetate paper is compared with that of the standard prepared. Apart from testing the physical-chemical properties of rubber closures, we also need to test for the functional properties. The first being the penetrability of rubber closures. It is important to test for the penetrability as this will affect fragmentation and self-sealing. These 2 properties will be discussed further in the presentation. So what is penetrability? It is the ability of the needle to enter the target surface of the rubber. The rubber closure cannot be too easily penetrable as this will cause the leakage of contents, and it also cannot be too resistant to penetration as this will be difficult for the needle to enter the rubber closure. The following are the steps for the penetrability test. First is to fill the 10 vials with water R. Then secure the vials with rubber closures. And finally, to pierce the rubber closure with a long beveled hypodermic needle perpendicularly to the surface. The force is measured, and the force required to pass the penetrability test will be less than 10 newtons. Deviation of the force measured between replicates should not be more or less than 0.25 newtons.
Moving on to our next property, which is self-sealing capacity. Self-sealing capacity is the ability to perfectly reseal after being punctured and after withdrawal of needle. It depends primarily on the elasticity of the rubber closure. It is important that the puncture must close so as to prevent entry of microorganisms from outside of the valves and leakage of contents of the vial itself. Therefore, it maintains sterility and integrity of the preparation. Pharmacopoeias mentioned that only multi-dose preparation need to be tested for this. Why? This is because single-dose preparations are usually disposed after a single penetration. Therefore, it is not so crucial. So how the test is done is to prepare a vial containing water R and the rubber closure to be tested. Using hypodermic needle, make 10 punctures on the rubber closure, piercing each time at different site. The vial is then immersed in methylene blue, and the external pressure is reduced for 10 minutes. Following that, atmospheric pressure is restored and maintained for the next 30 minutes. A good rubber closure should be able to reseal itself and prevent entry of methylene blue. Therefore, out of 10 replicates, none of the vials should contain any trace of colored solution.
The last test for functionality is fragmentation or coring. Fragmentation happens when small parts of the rubber closure are dislodged by the needle during piercing or because of abrasion. These fragments can be deposited into the injection solution, where there is a risk of injecting them into the patient, which can lead to toxicity and embolism. This is the method for testing for fragmentation as recommended by the USP and European pharmacopoeias. First, fill 12 clean vials with water to 4 ml less than the capacity, secure with rubber closures and allow to stand for 16 hours. Then, inject 1 ml of water into each vial while removing 1 ml of air. Do this 4 times for each vial at different locations on the rubber closure. Then filter the liquid from all 12 vials through a single filter with pore size less than 0.5 microns. Lastly, visually count the number of rubber fragments retained on the surface of the filter. To pass the test, there must not be more than 5 fragments visible.
Okay, so now we move on to the biological properties of the materials. It's important to take note of the fact that these materials may actually react with the body because the things that they are made with are actually very reactive substances. So it's important to establish that they do not actually react to the body. There are in vitro and in vivo tests. The in vitro test, as the name suggests, is actually carried out on cell cultures. In this case, human fibroblast cell cultures. There's the agar diffusion, direct contact, and elution tests. The agar diffusion and direct contact tests are actually more or less the same in the sense that both of them involve the cutting up of the material and placing it directly onto the cell culture. And then you basically see if the cells actually react to the material. Direct contact test is actually for materials which are not so dense, which means that when you put it on top, you won't squash the cells. And the agar diffusion test is for materials which are dense enough to definitely squash and kill the cells. So you need to put an agar layer above it to cushion the impact. The elution test works a bit like a Paul plate method. So you basically take the material extract and you mix it with the cell media and then you plate it. So you can use different combinations of these tests to give you the desired results. And after 24 hours, the cells in these tests are actually examined by the microscope to see if they're compatible with the material. So these are the grades of biocompatibility. As you can see, for a material to pass this in vitro test, it cannot go above a grade 2 reactivity, which is a mild reaction. So not more than half of the cells should be swollen, and there should be no extensive cell lysis. So the in vivo test is usually done on mice and rabbits. Systemic injection tests and intracutaneous tests. They both involve the material extract being injected to the animal body. For the systemic injection test, the material extract is injected into systemic circulation of mice, and the material passes the test if the mice do not show an extensive reaction to the material, such as convulsions or death. The intracutaneous test involves injecting the material into the skin of a rabbit. And for a material to pass that test, the rabbit should not have any severe reactions as well, such as erythema or edema. Like in this rabbit over here, you can see that it has severe erythema, which of course the material will fail. One thing to take note of is the fact that unlike other plastics or polymers, elastomers can pass the test. Elastomers can pass biological testing either via in vivo or in vitro method, which means to say that if it fails in vitro method, it can still pass if it passes the in vivo test. So either way, that's how it works for elastomers. So in conclusion, we've gone through the importance of why we have to test the materials for these sort of properties. And we can see that we've done the tests and standards for so far the physical, chemical, functional, and the biological properties of elastomers. These are actually very important because we want to determine the efficacy, and we want to make sure that the drug is efficacious when we are using this material for storage, and also to make sure that this material will not react with the human body when it comes to contact with it. So these are the references that we've used so far in this presentation. Thank you very much.